The process of photosynthesis has been divided into two different types, that is the light reactions and the dark reactions. Now, when we talk of the light reactions are actually light dependent reactions, and they involve light driven electron and proton transfer and take place in the thylakoid membranes. They produce the products of the light reaction. When we study, it produces ATP and NADPH. Both of these are used as energy currencies. Now, in the dark reaction, we have classified this as dark reaction because this process does not require light, or in other words, it is not directly dependent upon the light. So, therefore, the name dark reaction. And this process involves, or during this process, there is fixation of carbon dioxide into carbohydrates through a cycle which is known as Kelvin-Benson cycle. Now here, this dark reaction uses, or the reaction which is taking place during in the absence of light, utilizes the ATP and the NADPH produced during the light reaction, and this takes place in the stroma of the chloroplast. So we'll be studying over here the light reaction and the dark reaction to completely understand the process of photosynthesis in plants. Now, coming to the light reaction, I hope you have understood the difference between the light reaction and the dark reaction. Now, the light reaction is also known as the photochemical phase. So, we also refer to this as photochemical. Why? Because the photon is photons, that and chemical is the series of chemical activities or the changes which are taking place for the production of NADP and ATP. So photochemical phase, this is. Now over here, before we start the light reaction, we have to understand the formation of the light harvesting complex. Let us try and understand what is meant by light harvesting complex, or LHC. We have to understand this, and also the photosystems. Photosystems are the systems, or the mechanism by which the light is absorbed. So the light harvesting complex is having the PS1, that is photosystem 1, and PS2, that is photosystem 2. Now, let us try and understand what is meant by that. Now, we know that chlorophyll A is an important pigment which absorbs the light. So, chlorophyll A is the main center, and it has several other pigments. This is surrounded by several other pigments. These pigments can be chlorophyll B, C, D, E, or carotenoids or xanthophylls. So chlorophyll A is surrounded by accessory pigments. And all of them, like chlorophyll A, B, C, not A, but B, C, D, E, or the xanthophylls, carotenoids will fall under the accessory pigments category. Now, the chlorophyll A is the reaction center. It is the reaction center. Now, what happens is, when the light energy is, or when these are exposed to light energy in terms of photons, This light energy is absorbed by the accessory glands, accessory pigments, and these are then passed on to the chlorophyll A. As a result, chlorophyll A becomes excited and it releases two electrons. That means two electrons are released due to the energy which is being provided by the due to the absorption of the photons by the accessory pigments, which is finally passed on to the chlorophyll A, and which in turn gets excited and releases two electrons. Now these two electrons have a acceptor. These two electrons are having an electron acceptor. So this entire is known as light harvesting complex, because it is utilizing the photon, that is the energy, and in turn excites the accessory pigments. This energy passes to the chlorophyll A, and which in turn gets excited and releases electrons. Now the LHC, that is the light harvesting complex, is therefore made up of hundreds of pigment molecules, and out of which it has chlorophyll A, which is the reaction center. This entire light harvesting system is also known as antenna. This is also known as antenna complex because it is trapping, or it is harnessing the energy from the photons, and these are accessory pigments. Now the single chlorophyll A molecule forms the reaction center. Again, this reaction center has been classified depending upon the absorption of certain wavelength of light. It has been classified as PS1 and PS2. PS1 is photosystem 1 and PS2 is photosystem 2. Now in photosystem 1, the reaction center, that is chlorophyll A, absorbs light of wavelength 700 nanometers, whereas in PS2, the chlorophyll A, in both the cases, the reaction center will remain the same. The accessory pigments will also be similar, but they differ mainly in the absorption, the wavelength absorption. So over here the PS1 will have 700 nanometers and PS2, the reaction center, will absorb wavelength of 680 nanometers. These two photosystems differ this way, and therefore PS1 can also be referred to as P700, or it can be referred to as P680. Whether we refer PS1 as P700 or PS2, that is photosystem 2, as P680, both are considered correct. Now, in laboratory conditions, if we take chlorophyll A separately, surrounded by the accessory accessory pigments, and due to the absorption of light energy, the molecules, accessory pigments, will absorb the light and will pass the light or this energy to chlorophyll A, and this electron received will be removed from the shell, and since in the laboratory condition there is no acceptor, it will return back to chlorophyll A, and while returning back it will emit energy of certain, or in the form of color of certain wavelength, and this process is known as fluorescence. This is in the laboratory condition when we are extracting only a molecule of chlorophyll A and is allowed to exposed to sunlight, then it will get excited, and during excitation it will release two electrons. But since in the laboratory condition there are no electron acceptors, this electron will come back to chlorophyll A molecule, and while coming back to its ground state, it will emit energy or light of certain wavelength, and that process is known as fluorescence. But over here in the actual situation, when the electrons are released by chlorophyll A due to the excited state, there will be an electron acceptor, and this, depending upon the chlorophyll A, what wavelength of light it is absorbing, we have PS1, that is having the 700 nanometers as absorption level at the absorption wavelength, and PS2 at 680. So here we are going to understand the PS1 and PS2. Now this photosystem, both the photosystems are linked together. These photosystems are linked or are joined by the electron transport system. So when we study over here, we find that the electron transport system, or the ETS we call it, it is also referred to as ETC. It is actually consisting of cytochromes, and cytochromes are actually having proteins. They are proteins and having a cofactor, and they act as electron acceptor. So over here the cytochromes will function for as an electron acceptor, and the movement of electrons take place. So over here if we see the PS1, after the light has been absorbed, this is light harvesting complex. Now after this is PS2 we are talking. So the light, it is again the light harvesting complex consists of chlorophyll A and the surrounding accessory pigments. Over here on the light is focused, then the electrons are released, which are taken up by the primary electron acceptor. Now this primary electron acceptor, it is having at 680 nanometers, and here the photolysis of water takes place, which results into the electrons plus water molecule plus nascent oxygen. This is the lysis, that is the water, this hydrolysis or photolysis of water takes place due to the light, and over here the water molecules on breaking produces electrons, hydrogen and oxygen. So this oxygen is one of the products of photosynthesis and is released by the water. Now this process of the electron acceptor, over here the passes the electron to the series of reaction which we call it as ETS or ETC, and here ADP gets converted into ATP. Now since there is phosphorylation, we call it as, this process is known as photophosphorylation. There are different types of photophosphorylation which we'll be studying in subsequent video, but over here after the light has been absorbed by the light harvesting complex at 680 nanometers, that means we are talking of PS2 system, and the splitting of water molecule which produces oxygen as the product of photosynthesis, the electrons are released which are taken up by the ETS, that is through the pigment cytochrome. It is B6F, and this over here, the electrons are not used up, but they are passed in a series of reaction, which results into the production of ATP into ADP. And here, from these, the electrons further enter into the LHC and also the light energy, due to which again it becomes excited, and here the primary acceptor takes place from this at P700 nanometers, and therefore this is PS1. So the electrons are accepted over there, and as a result of this there is formation of or the addition of these electrons which are produced due to the excitation of P700 center, reaction center. The NADP gets converted into NADPH. So the products of the Z scheme, because it is representing a Z shape, this is known as Z scheme of light reaction, and it produces ATP and NADPH. So the whole system of the transfer of electrons, starting from the PS2 to the primary acceptor to the electron transport system, where only the electrons are being passed, and they are then the energy produced is utilized for conversion of or photophosphorylation of ADP into ATP takes place, and the electrons are passed, and in turn when LHC or the light harvesting complex absorbs the light of 700 nanometers, get excited and pass to the primary acceptor of PS1, from where it reduces or it converts NAD into NADPH, and this is why it is known as Z scheme. So over here the shape is obtained when all the carriers of electrons are placed on a redox potential scale. Also, when the protons which are being produced during the process are released within the lumen of the thylakoid, whereas the oxygen which is formed is released towards the outer side of the membrane. So this completely covers or relates the PS1 and the PS2 system of the light reaction, which are very important, and produces the ATP and NADPH, which are being then utilized as energy source for the dark reaction or for the fixation of carbon dioxide into carbohydrates.